Welcome to this video. Streptococci are among the most important bacterial pathogens affecting both humans and animals. They are responsible for a wide range of diseases including mastitis, respiratory infections, septicemia and meningitis. Because many Streptococcus species appear similar under the microscope, accurate identification requires a combination of laboratory techniques. In this video, we will explore the key methods used to identify and differentiate streptococci in a laboratory. We will look at direct microscopy, culture characteristics, hemolysis pattern, Lancefield grouping, and important biochemical test. By the end of this video, you will understand how microbiologists distinguish different streptococcal species and how these techniques help in the diagnosis of infectious diseases in veterinary medicine. Streptococci are a diverse group of gram-positive cocci that play an important role in both human and veterinary infections. To identify and differentiate different species of streptococci in the laboratory, microbiologists rely mainly on two important characteristics. First, the type of hemolysis produced on the blood agar, which reflect how the bacteria interact with the red blood cell. Second, the Lancefield grouping system, which classifies streptococci based on the specific carbohydrate antigens present on the cell wall, that is the cell wall antigens. Together, these features provide the essential starting point for the laboratory identification of the Streptococcus species. First, we will look into the hemolysis pattern. It is one of the most important characteristics used in differentiating streptococci. Hemolysis refers to the breakdown of red blood cells in the culture media surrounding the bacterial colonies. There are three major hemolysis patterns that is observed in streptococci. The first one is beta hemolysis. It produces a clear transparent zone around the colony due to the complete destruction of the red blood cells. Here in this picture you can see beta hemolysis. In the below picture, the center one is the beta hemolysis. You can see a clear transparent zone around the colony. Next is alpha hemolysis. It causes a greenish discoloration around the colony, this left one. This occurs because of the partial hemolysis and oxidation of the hemoglobin. The last type is the gamma hemolysis. Despite the name actually means there is no hemolysis occurs and the blood agar surrounding the colony remain unchanged. The right one, this is the gamma hemolysis, which means there is no hemolysis. Among the different hemolytic patterns, the beta hemolytic streptococci are considered the most pathogenic streptococci, especially in animals. These organisms produce complete lysis of the red blood cells, creating a distinct clear zone around the bacterial colonies on the blood agar. In veterinary medicine, a classic example is Lancefield Group C streptococci from horses, which typically produce large zones of clear beta hemolysis. Observing these hemolytic patterns provide an important clue during the initial identification of the streptococcal isolates. In addition to the hemolysis, streptococci can also be classified using the Lancefield grouping system. This system is based on group-specific carbohydrate antigen located in the bacterial cell wall, often referred to as the C substance antigen. Using this system, the streptococci are categorized into multiple groups, including group A through H and K through V. However, not all streptococci possess these antigen. Therefore, some isolates may not be groupable under this Lancefield grouping system. The traditional method used for Lancefield grouping involved the extraction of the C substance antigen from the bacterial cell wall. The antigen can be released using autoclaving or by acid extraction with hydrochloric acid. The extracted antigen is then tested using a ring precipitation test, where the antigen solution is layered over the antisera containing the antibodies specific for the different Lancefield groups. If the antigen reacts with a particular antiserum, a visible precipitate line will form, indicating the corresponding the Lancefield group. So, like this, we can classify different streptococci species. The next method for the Lancefield grouping is the lattice agglutination test. Today, the lattice agglutination test is the most commonly used method for Lancefield grouping in diagnostic laboratory. In this method, the lattice particle will be coated with antibody against specific Lancefield antigens and are mixed with a bacterial extract. If the corresponding antigen is present, we can see visible agglutination or clumping occurs within seconds, and commercial kits are widely available for identifying Lancefield group A, B, C, D, F and G, making this technique rapid, simple and highly practical for routine laboratory diagnosis. Important Streptococcus species in veterinary medicines. There are several species of Streptococcus that is important in veterinary medicine. These organisms are classified mainly based on the Lancefield serological group or the type of hemolysis it produced on the blood agar. Understanding these characteristics help veterinarian and microbiologists quickly identify the organism responsible for the disease. These are the important species like Streptococcus pyogenes, Agalactia, Disgalactia, Equi, Suepidemicus, Suis, Uberis or Streptococcus canis. The first important species is Streptococcus pyogenes, which belong to the Lancefield group A and it produces beta hemolysis. But it primarily affects humans, causing diseases such as scarlet fever and streptococcal pharyngitis. Another important species is Streptococcus agalactia, which belong to the group B Lancefield group. It usually produces beta hemolysis, although alpha and gamma hemolysis may sometimes occur. It mainly affects cattle, goat and sheep, where it is an important cause of mastitis, but it can also affect humans and dogs. Several important veterinary streptococci belong to the Lancefield group C. The first one is Streptococcus disgalactia, subspecies Disgalactia. It mostly infects cattle and lamb, produce mainly alpha hemolysis, although other hemolysis patterns may occur like beta or gamma. Another subspecies, that is Streptococcus disgalactia subspecies Equisimilis, usually produce beta hemolysis, which infect a wide range of animals including horses, pigs, cattle, dogs and birds. The Streptococcus equi subspecies Equi is a well-known pathogen of horses causing the disease strangles. It also shows beta hemolysis. Closely related is Streptococcus equi subspecies Suepidemicus. It infects horse, cattle, pigs, poultry and lamb. It also produces the beta hemolysis. That is the Streptococcus, that group C Streptococcus that infect the horses. All three of them will form beta hemolysis. But the Streptococcus disgalactia subspecies Disgalactia that affect the cattle and lamb, it shows usually alpha hemolysis. Now we will learn about the group D, E and G. Some Streptococci belong to Lancefield group D, including the species are now classified as Enterococci, such as Enterococcus faecalis and Enterococcus faecium. These organisms can infect many animal species. It typically produces alpha hemolysis. Another group D Streptococci is Streptococcus galolyticus. It also shows alpha hemolysis, which infects birds and humans. Next, we will learn about the group E Streptococci, that is the Streptococcus porcinus. It shows beta hemolysis and it's mainly associated with the diseases in pigs. Finally, the Streptococcus canis, it belongs to the group G, producing beta hemolysis and affects dogs and cats, and it occasionally causes mastitis in cattle. Other important Streptococci, the first one is Streptococcus suis. It is an important pathogen of pigs. It producing alpha hemolysis and it is in the Lancefield group D, and it can also infect humans. Some of the Streptococci are not assigned to a Lancefield group. The first one is Streptococcus uberis. Is another significant veterinary pathogen that causes mastitis in cattle, and it typically shows alpha hemolysis. Finally, Streptococcus pneumoniae is a very well-known alpha hemolytic streptococci, and which infects both humans and also animals like horses and also rodents, particularly causing respiratory infections. Laboratory identification of Streptococci involves several important steps. These include direct microscopic examination, isolation and culture on suitable media, evaluation of the colony morphology, and performing biochemical and presumptive identification tests. Together, these methods allow accurate identification of the streptococcal species that is responsible for infection in humans and animals. First, we will look into the direct microscopy. In direct microscopy, the smears are prepared from the clinical samples such as pus, exudate, centrifuged milk deposits, or urine sediments. The smears are fixed and stained using the gram staining procedure. What we will find under the microscope, the streptococci appear as a gram-positive cocci that will be arranged in chains, especially when observed from broth culture or infected tissues. And the organism Streptococcus pneumoniae, it will appear like a pair of cocci, that is diplococci. Next is the isolation and culture. The streptococci are considered fastidious organisms. It means they require enriched culture media for their growth. They don't grow in the normal nutrient agars. They grow best on media supplemented with blood or serum. And the growth of many species is enhanced in the carbon dioxide enriched atmosphere. These bacteria will ferment glucose and other sugars, but importantly, they do not produce gas during fermentation. Several culture media are used for the isolation of streptococci. The routine laboratory media include sheep blood agar, ox blood agar, selective blood agar, and Makongi agar, that is without crystal violet. For mastitic investigation, the milk samples may be inoculated on blood agar, Edwards medium, and Makongi agar. In case of herd mastitis investigation, the milk sample collected from the infected animal are cultured to identify the causative bacteria. The mastitic milk samples are commonly quadruplated on blood agar plates. This technique helps in isolating different bacterial species that is present in the sample. The blood agar may contain 0.1 to 0.05% esculins, which allow detection of esculin hydrolysis and important biochemical features that is used for the bacterial identification. After inoculation, the culture plates are incubated aerobically at 37 degrees Celsius for 24 to 48 hours, allowing sufficient time for the bacterial colonies to grow and can be identified. Most streptococci produce small colonies, typically around 1 mm in diameter, after 48 hours of incubation. In beta hemolytic streptococci, the colonies usually appear translucent in blood agar, and colony variation can also occur among streptococci. Mucoid colonies are sometimes observed in organisms like Streptococcus equi subspecies, Streptococcus equi subspecies Suepidemicus, and Streptococcus pneumonia. These mucoid colonies are usually associated with the presence of capsule that contribute to the bacterial virulence. The special colony morphology of the pneumococcus or the streptococcal pneumonia, it produces either a mucoid colony or a distinctive colony type known as the Trotman colonies. These colonies are typically flat with smooth borders and a central concavity, resemble the shape of a Drotsman piece. The basic identification test. When these colonies are examined microscopically, we can detect gram-positive cocci, and another major identification test is the catalase test. Streptococci are catalase negative, which help to differentiate other gram-positive cocci like Staphylococcus, which are catalase positive. Biochemical identification of bacteria can be done using commercial kits or conventional tests. The commercial kits are very easy and rapid to use, providing multiple results at once. The conventional biochemical tests are traditional method that detect the metabolic activities like sugar fermentation. The common media we are using is peptone water sugars with serum and cysteine trypticase agar with carbohydrate disk, which help to assess bacterial utilization of specific sugars. These methods are essential for accurate bacterial identification. Several presumptive identification tests are used in microbiology laboratory to differentiate important species of streptococci. First, we look into the group A streptococci. Group A streptococci are susceptible to bacitracin, which makes the bacitracin disk test useful for their identification. However, they are negative for the CAMP test, hippurate hydrolysis, Optochin susceptibility, and the bile solubility. So, group A streptococci positive for bacitracin test. Next, we will look into the group B streptococci, such as Streptococcus agalactia. It shows positive for the CAMP test and also hydrolyze sodium hippurate. However, they are resistant to bacitracin and negative for Optochin susceptibility and also for this bile solubility. In contrast, Streptococcus pneumonia can be identified by its susceptibility to the Optochin and also bile solubility, both of which are positive in the case of Streptococcus pneumonia. Actually, these steps help to distinguish this Streptococcus pneumonia from other alpha hemolytic streptococci. Next, how we will differentiate the group C streptococci, that is equine, that affecting equines. The equine group C streptococci can be differentiated using carbohydrate fermentation test, particularly with sugars like trehalose, sorbitol, lactose and maltose. Streptococcus dyscalcius subspecies equisimilis, it will ferment trehalose and maltose but does not ferment sorbitol. Okay, but the lactose fermentation may show variable reaction in the case of equisimilis. Next is Streptococcus equi subspecies equi. It does not ferment trehalose, sorbitol and lactose, but it ferment maltose. So in the case of equi, it ferment maltose. And the last one is Streptococcus equi subspecies suepidemicus, which ferment sorbitol, lactose and also maltose, but few strains may show negative reaction for maltose. These biochemical reactions are important for distinguishing closely related group C streptococci isolated from the host in diagnostic laboratory. So we can distinguish the equine group C streptococci with the help of the different carbohydrate fermentation test. Enterococci and the group D streptococci can be distinguished by several physiological properties. They have ability to grow at 45 degrees Celsius and it tolerate 40% bile, and they also hydrolyze esculin. The Enterococci and the other group D streptococci we can distinguish using carbohydrate fermentation test and the salt tolerance test. The Enterococcus faecalis, which belong to the group D, it ferment lactose, sorbitol and mannitol but does not ferment arabinose. It also grows in 6.5% sodium chloride, which is a characteristic feature for the Enterococci. Next is Enterococcus faecalis, which belong to the group D. Lancefield classification, it ferments lactose, arabinose and mannitol but does not ferment sorbitol. Like other Enterococci, it can also grow in 6.5% sodium chloride. All the Enterococcus species will grow in 6.5% NaCl. The Enterococcus durans, which will ferment lactose only, and it does not ferment any other sugars, and also shows growth in high salt concentration. The last one is Enterococcus avium. It group Q, ferment all 4 sugars: lactose, arabinose, sorbitol and mannitol, and also grow in 6.5% sodium chloride. Finally, the Streptococcus equinus is a group D streptococci, show variable reaction to the lactose, arabinose, mannitol fermentation and does not ferment sorbitol. It is a clear negative for the sorbitol. For others, it is variable and it cannot grow on 6.5% sodium chloride. There are several presumptive identification tests are used in the laboratory to differentiate important streptococcal species. These include CAM test, hippurate hydrolysis, pigment production, Bacitracin susceptibility, Optochin sensitivity, and bile solubility testing. CAM test is used for identifying group B streptococci, particularly the Streptococcus agalactia. In this test, a culture of the Staphylococcus aureus is streaked across the center of the blood culture plate, and the suspected streptococci is streaked perpendicular to it. A positive CAM test produces a characteristic arrowhead-shaped zone of enhanced hemolysis. So another important test to distinguish the group B streptococci is hippurate hydrolysis, and which help to identify the group B streptococci. And group B streptococci also may produce orange to red carotenoid pigments when incubated anaerobically on a specialized medium such as GBS occur. And that's all for today. Thanks for watching.